Welcome to Chromosomes Part B, the second lesson in our little two-part series on chromosomes. In this lesson, you're not going to learn a great deal more about chromosomes themselves, but what you are going to learn about is a technique for analysing chromosomes, and through that, you may also learn some things about biology and how biology is used. And the topic is karyotypes. A karyotype is a graphical representation, a picture really, of all the chromosomes in a cell. It's not a diagram. A karyotype is not a diagram like we would make a graph in science, and it's not even an illustration. It is actually a photograph, but it's a photograph that's been manipulated. So the photos are taken of somatic cells, that is the body cells, the cells with 2N ploidy, during cell division. That's the only time, well, that's the time that they're going to be best seen in their X shape. And once those photographs have been taken using a microscope, then the scientist will then literally cut and paste the X shapes and move them around on a screen until they are lined up. And they're lined up in order of descending size. So starting with the largest chromosomes and moving on to the smallest. Chromosomes are very consistent in their shape and their size and some other features as well. And they're so consistent that they can be coded with numbers and letters, and a geneticist can recognize exactly which chromosome is which out of any cell in any human. Before we have a look at some examples of karyotypes, we also need a new concept. Or it's not an entirely new concept, but it's one that we have begun to discuss in previous lessons, but we haven't fully covered it. I've already said that chromosomes are very consistent, so consistent in shape and size and their banding pattern, that's how they look under a microscope, that a competent geneticist can find a chromosome 7 or a chromosome 13 or a chromosome 1 in any somatic cell. But remember that these are 2N cells. They're somatic cells. They've got a ploidy of 2, so they have 2 sets of instructions, which means that every cell will have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, etc. One of those chromosome 1s came from mum and the other one came from dad. One of those chromosome 15s came from mum and the other one came from dad. And so in making a karyotype, the geneticist will find both of the chromosomes 1s and will stick them next to each other, will match them up on the screen. And those matched pairs are called homologous pairs. Each of the individuals is known as a homologue. And as I've already said, one homologue comes from mum and one comes from dad. Together, they make up an homologous pair. To get back to our book analogy and the critical and ever-present issue of information, those homologous pairs you can think of as being 2 pages which have different instructions but about the same thing. Now that might seem like a contradiction, but think about this. Imagine that homologue 1, that is, let's say we're talking about chromosome number 3. Remember that the cell that we're interested in will have 2 chromosome 3s. One homologue will come from mum and one homologue will come from dad. And on those chromosomes will be a whole bunch of instructions about how to make a human being or to keep a human being running. So let's take a really simple situation, and on this, in this homologous pair, homologue 1 gives instructions to the cell and therefore to the body to make the eyes of that person blue and make their hair red. But homolog 2, although it will definitely have instructions for eyes and will definitely have instructions for hair, it may not say the same things about the eyes and the hair. So homolog 2 might say, make the eyes brown and make the hair black. So now we have conflicting instructions. We've got two sets of instructions, but they don't about the same thing, but they may not give the same instructions for those things. And that, I hope you can see, introduces a very interesting dimension to understanding genetics and understanding how parents have offspring who look similar to them but not the same. Well, I've talked a lot about karyotypes, and here actually is a karyotype. There you can see a one homologous pair. That's the homologous pair for chromosome 5. So one of these came from mum and one came from dad. You'll also see that the scientist has drawn in some dotted lines. The dotted lines obviously aren't floating around in the cell. They've been put in after the fact, and they just show where the centromeres are. If you recall, the centromere is a piece of protein which holds the two chromatids together. Well, I have a question for you to ponder. If these are all chromosomes, why are there no X shapes? Now, let's take this example here, chromosome number 3. Don't be fooled into thinking that we have an X shape which is kind of separated. In fact, this is one whole chromosome and this is one whole chromosome. You've got two chromosomes, and my earlier statement should mean that there are two X shapes. So why are there not? Well, the answer is that they are X shapes, but the two legs or the two chromatids are so close together that you can't see them as an X. If you look very carefully at the top of chromosome 3 here, on the left-hand side, the homolog on the left-hand side, you'll see they're slightly rounded with a dip in the middle. That's because the chromatids are lying so close together that you can't see the X shapes. But they are chromosomes, and they do have, each one of these, all the ones on the page, have two chromatids. All right, well, we've already said that chromosomes code different information to get the cell to do different things. So each chromosome out of the set of 23 has a different job to do. In fact, it has many different jobs to do. We have approximately 25,000 genes or 25,000 individual instructions, and those 25,000 instructions are crammed onto only 23 pages. So if you do a little bit of division, that means on average you expect to have about 1,000 genes, that's 1,000 instructions for each page of genetic information. And those instructions could be about very, very different things. Like we've already seen, you could have eye color and hair color on the same chromosome. Well, there is a couple of chromosomes which have the job of giving the organism its sex, making it a boy or a girl. Humans have two kinds of sex chromosomes, and I'm sure most of you have heard of the X and the Y chromosome. But all the other ones are called autosomes, and they don't determine sex. Now, please note that sex chromosomes do have information for other features apart from making you a boy or a girl. It's just that autosomes give no information for making you a boy or a girl. So I'll say that again, a sex chromosome determines whether you're going to be a boy or a girl, but it also has other information on it which is unrelated. Autosomes have all sorts of other information but have no influence on whether you turn out to be a boy or a girl. Humans have two sex chromosomes and they have 22 autosomes. So let's have a look at this on a karyotype. There are the sex chromosomes there. So you'll notice if we go through our numbering from largest to smallest, 1, 2, 3, 4, 5, all the way through down to chromosome 22, or the 22nd pair, and the 23rd homologous pair is, in this case, one X and one Y. So what would be the sex of this person? Well, I'm sure most of you would know, if not all of you are very well aware, that this would be a boy, because a boy has to have one X and one Y. To be a girl, you would have two X's. There would be no Y's there. Well, remember, the karyotype is a diagram. It's a photograph which has taken from a dividing cell, and the chromosomes have been split up on a screen and shifted around until they're all lined up in homologous pairs. And just as we saw on the previous page, there should be 46 chromosomes. Now, males will have, will be 2n equals 46. That's going to be the same for everyone, with 44 autosomes. And then their last homologous pair, the 23rd homologous pair, will have one X chromosome and one Y. For girls, they will still be 2n equals 46. They will still have 44 autosomes, non-sex chromosomes, and then they will have one X and another X. So every human being has one X chromosome, but boys have an X and a Y, and girls have an X and another X. That number is very important. As soon as that varies, then problems start to occur. You think that having more information would be a good thing. I don't know why such serious consequences follow, but in humans, at any rate, having extra chromosomes is a big deal and is often lethal. The infant will die before birth, and for those where the condition is not lethal, severe defects are easily apparent. And in fact, that's one of the reasons why karyotypes are done, is to work out, say a child is born and appears to be having difficulty learning. So they might do a karyotype on that child and realize that they have an extra 21st chromosome. So instead of having a homologous pair, you have that child has three, not two, but three chromosome 21s. If that was the case, then they would be called a Down syndrome person. So Down syndrome is caused by having an extra 21st chromosome. All right, so that's the end of our karyotypes lesson. Again, not much new about the theory of chromosomes themselves, but a practical application of science and genetics in working out the kinds of problems that may arise, or, of course, whether there is no problem at all. If you want to get some more information, you can read your Pearson textbook, page 332, and there's a couple of questions there. All right, that's us done for this lesson.